Our next speaker is Professor Shino Suzuki. Professor Suzuki leads a research group focused on extremophiles and the limits of life. Professor Suzuki is an associate professor at the Japan Aerospace Exploration Agency and also a visiting senior researcher at the Japan Agency for Marine Earth Science and Technology. Professor Suzuki is going to speak about subsurface microbiology, astrobiology, and the evolution of life. Please, Professor Suzuki.
Thank you. So today I would like to talk about life in the Serpentinized setting, implication for metabolic strategy of early life. So yes, so what was going on? So the 4.6 billion years ago, Earth like this without life, but now the Earth with a full of life. So during this point, what was going on? This is a long time question for human being, but it has not been answered yet. So here is life in Earth called evolution. So life has been evolving and adapted to the drastically changing environments like this. So especially it's about 2.4 billion years ago, the great oxidation event happened. And then this is really because of the O2 producing photosynthesis bacteria. So that this event changed a lot drastically the Earth's surface, as the other speaker also talked. So then, but before the great oxidation event, the environment must be more reductive environment. So and the life when life started about 4.0 billion years ago, the environment must be a really reductive environment. So the sulfur, the evolution of life. So now we know about the current microbial life, almost. Then current microbial life is evolved. The evolutionary strategy is like diversification and complication. These occur like gain and loss function, or genome streamlining, or in the sympathetic theory. And we start knowing that the strategy of how the evolution can occur on the Earth. And also we try to understand the chemical evolution and primordial life. The primordial life and chemical evolution are still like we have very limited information, but start knowing like from the genomic data and of the primordial life, and also chemical evolution at the various lab experiments. But still between the chemical evolution and primordial life, we have a huge gap, and this is the origin of life. So how can we address the origin of life? So that is, so in the biological approach, everybody try to understand the primordial biological system from the present life, and also from the understanding from the chemical evolution. So illustrating the function of primordial biological system is the key to connect between chemical evolution and present day life. So the life on the Earth, the current Earth, the life are spotted the solar energy and also geochemical energy. But the mostly solar energy, 99.97% is like a huge amount of energy is supported by the solar energy. So photosynthesis supports most of the ecosystem on the current life. So the ecosystem in current Earth is mostly supported by photosynthesis, even if the deep subsurface ecosystem. So here is the deep subsurface sediments. So the organic from the photosynthesis is transported into the deep subsurface, and ecosystem in the deep subsurface is driven by the like lignin-like compounds. So that those kind of compounds are really difficult to degrade, even if the surface, but in the long time, in the geological time, the lignin, or the really difficult compound from the photosynthesis, synthesized by the photosynthesis, is like the are degraded in this ecosystem, and that support the ecosystem in the deep subsurface. And also, even if it's hydrothermal vent, this is the reductive compound that is produced from in the deep, but oxidative compound comes from the surface. Then oxidative compound is also the product of photosynthesis, and that at least affected by the huge largely affected by the oxygen. So then between the reductive compound and oxidative compound, the oxic and anoxic interface create a huge disequilibrium between these two, and the microbes and life can utilize the power between these two. So what are the ecosystems without photosynthesis? So here is a low powered life. I've been working on the Cedars serpentinization site for 9 years and for more than 10 years. And then here is the peridotite rocks and exposed at the surface. And we can see a lot of such a beautiful place, the calcium carbonate rock at the surface because of the serpentinization. So the serpentinization so the serpentinization on Earth is like this. So the peridotite is the name of the rock making up the top layer of the Earth's mantle below the crust. And the peridotite is composed with the two main components, like olivine and pyroxene. So this rock react with the water and then it makes the serpentine. So during this reaction, a couple of different reaction occurs. So at the end of the reaction, so the hydrogen is produced and also hydroxide ion. And if the dissolved CO2 is present, methane also can be product. So the hydrogen is a really good energy for life, and methane could be a source of the organic carbons and also energy for life. So the materials, compound could support early life. So the geochemical property of serpentinized water discharged from the Cedars serpentinization. So here is the water coming up from the Cedars. So the pH is really high, between pH 11 and 12, and the EH is low. It's about minus 700 millivol to the minus 550 millivol. It is one of the lowest records of the current as far as I know, and also very rich in hydrogen and methane, and relatively rich in calcium, iron, and also poor in the dissolved inorganic carbons and poor in electron acceptors like oxygenic compound like oxygen nitrate and sulfate. So those are the builder detection limit. So serpentinization is such serpentinization site is really reduced. Such reducing environment is considered as the analog of early Earth or in Mars. So the Cedars, the diagram of Cedars is like this. So the water comes deep and react with the rock and make hydrogen and methane and those kind of stuff deep. So and support life in the water. So the water is organic carbons for the photosynthesis can be degraded at the early stage with the oxygen, and then most of the oxidative compound can reduce during the process. So the serpentinization setting is mostly isolated from the effect of photosynthesis, not completely, but so here is so therefore we can call it like an earlier analog. So chemical and physical constraint and driving force and the biological function should be similar to the earlier setting. So then the biological metabolism adapted and optimized to the analog are earliest to reflect the biological metabolism that would likely to occur there. So yeah. So the life always adapt to the environment. So that we cannot see the really all ancient life at this site, but the constraint, if the constraint and driving forces are quite similar, then we can see somehow the like biological metabolism that was likely to occur there. So the serpentinization site on the Earth right now is like this. So there is a lot of different sites, like terrestrial or seafloor serpentine hosted site are distributed on the Earth. So not only on the Earth, only in the Earth, but the potential serpentinization occur in the solar system. So first of all, the origin is really a common mirror in the solar system. So here is the rock from Itokawa that is returned by the JAXA's Hayabusa 1 project. And even if the asteroid, so the rock contain the olivine. So the olivine is contain the many in the meteorite as well. So we know that the olivine is really a common mineral in the solar system. So the Mars also orbital mapping suggested the presence of olivine and also potentially the presence of serpentine and carbonate. Those are the product of the serpentinization. So that the water lock reaction should be occurred in the past. So then in Enceladus is also the serpentinization H2 generation should have occurred in Enceladus. So though detected in the plume, they detected the hydrogen a lot and also a couple of organic molecules. So then they consider that the serpentinization and its generation should have occurred in the Enceladus. So the Cedars, the composition of the Enceladus ocean resembles a hybrid of Ikka Fjord, seawater, and velocity, and the sea that is really from Glenn's paper, the book. But here is the Enceladus ocean inferred, and the city is very close to the Enceladus ocean, the geochemistry. So in terms of the redox and pH. So none of these sites can capture all the essential features of the Enceladus ocean, but they exhibit at least one key similarity to the infogeochemical properties of the Enceladus plume. So the Cedars is the potential analog of yes, at the same time the Enceladus too in terms of the redox in the pH. So here is the springs at the Cedars serpentinization site I've already showed, but like there are over 100 springs in this site, and then we can collect a different samples like on this site. So I've been talking like the Cedars serpentinization could be related to the serpentinization could be related to the emergence of life, but actually this is kind of really difficult compared to the other like systems on the Earth like now, like more like a photosynthesis world. So the one of the difficult reason is that the nutrients. So the calcium, phosphorus is a very important CO2 and carbon and phosphorus are really important component for life. But these compounds can react with calcium easily, as you can see in the first the couple of the pictures of the cedars, but we see a lot of calcium carbonate at the surface because the carbonate it's really easy to react with the calcium and also the phosphorus too. So it's really difficult to get those nutrients to the inside of the cells. So then when they want to the respiration breathing, so they often use uh, proton motive force. So to make a gradient, the light microbes pump out the proton to make a gradient, but outside of the pH is really high, so it's very easy to neutralize. So that's the reason why it's difficult to make ADP, but for the respiration. And also at the same time, there's almost no electron acceptors, so microbes can neutralize the oxygen, nitrate, and sulfate, and those kind of stuff. But we don't detect those kind of stuff in this system a lot. So, and also pH homeostasis and protein holding could be difficult at the pH 12. So in this So when we think about as the habitat on the current Earth, it's really difficult, and it's really difficult to explain how to make a living. So we try to culture the organisms a lot, uh, and we wanted to culture actually the life in the deep subsurface organisms, but it was quite challenging. And uh, but anyway, like so, no culturable environment relevant microbe was not reported for us when we started the study, and from the Serpentinization setting. So we then first we could successfully isolate the environment relevant microbes that can live at the oxygen and anoxic interface. So here is A1B1H1, and we call it Serpentinimonas. So, but interesting things is that the water, as I mentioned, like does not contain not contain, but it does not a phosphate in the bicarbonate are not were not detectable. So were not detectable. So then that is one of the target how these organisms can utilize the phosphorus and bicarbonate in the system. So the Serpentinimonas distribute almost everywhere in the Serpentinized site. So and then when so probably at the interface, anoxic and anoxic interface. So microscopic observation Serpentinimonas strain A1 is like this. So when we grow with thousand, here is this white powder is a calcium carbonate. So then when we grow it on a calcium carbonate, we see like calcium carbonate change more like sticky part, and then we found that the microbes attached to the calcium carbonate. The green color is the microbes, so that the organism can utilize the hydrogen. Uh, then the green color is a microbe. So that we found that the organism attached to the calcium carbonate. So that this organism's optimum pH is 11 to 11.5, and this is actually when a couple of days ago I found that this was on the Guinness World Record as the most alkalization in the organism. So so here that the organism is like about optimum pH is 11 and 11.5, and electron donor is hydrogen, acetate, and others, and electron acceptor is oxygen, nitrate, chill sulfate, and so this is like chemolyso-autotrophic life, facultative chemolyso-autotrophic life, and utilize a rubisco to fix the carbon. So the carbon source is so when we grow is on the on solely carbon sources, the calcium carbonate, they grow well. So like this. So so we know that even if we cannot detect the bicarbonate in the water, but can utilize the calcium carbonate as a carbon source for this life. So not only the calcium, but also phosphorus also that can be a hydroxyapatite in the system at high pH and low pH environment. So so we think that this organ can utilize and hydroxyapatite, and that was true. So this organism really preferred to utilize a calcium phosphate or hydroxyapatite as a phosphorus source. So this organism utilizes hydrogen from rock water reaction, and also utilizes solid minerals like calcite and hydroxyapatite. So it's really kind of low power life. So, but we wanted to know that Serpentinum must grow at the interface of an oxic and oxic interface, but I really wanted to know like what kind of organism is living in a deep subsurface. So, but culturing deep organism is quite challenging. So employed the metagenomic strategies to get to know the information as a genomic information. So they all so first initially the microbes is just only we could know about the microbes only from the cultivated cells, But now, like getting better and better with the bioinformatics technologies, and then we see the genomic information from the metagenome and also metagenomic assemblable genomes, and now we can we can combine with, like, for example, the systems biology, like genomics transcriptomics, proteomics, and also sometimes we can combine with synthetic biology, or sometimes like AlphaFold too, like to know the function of the genes. So then when we combine the genomic information with a couple different strategies, then we start expanding understanding of life in the deep subsurface setting, especially the deep organism in the deep growing organism in the deep subsurface was quite difficult. So that this technology really opened the window or opened the door of the deep subsurface or the microbes. So then when we see the genomes of from the deep subsurface environment. So firstly, like as we expected, like we didn't see any no respiratory gene, no terminal electron acceptor reductase were detected in the metagenome of deep one. So then nobody can do like a breathing respiration in the system. So and then majority of the organ is like about 70% is called like a CPO, and independent life are mostly acidogen. So when we look at the community composition, so that 70% is CPO and 25% is of the acidogen. So mostly like these two are the major organisms in this system. So the CPO is recently described, not very recently, but 2015 and 2016, we start seeing the genomic feature of the CPO organism. So this group is a genetically diverse but physiologically unknown microbes, but occupy like more than 15% of entire like a diversity. So then a candidate file validation, the typical feature of the candidate file validation is that the lacking biosynthetic pathways, introns in the ribosomal genes, and also unusual ribosomal protein composition. So the CPO in the Cedars also lacking many genes required for mating life. So function in the pathway, most of the microbe have is like this, but basically the CPO here and also the side also, but have the duplication, translation, and transcription, and also cell wall synthesis, but less the like respiration and also the amino acid synthesis integration. Those lipids and those kind of genes are not encoded. So far, lifestyle is unexplainable only if the genes coded by the genome. But recently, you start knowing that this is a kind of episymbiotic organism. That the globe is the host, but it's not really sure how the life strategy is. So when we look at the microbes in the CPO on the Cedars, so we detected the CPO that is as an aggregate. So the green colored cells are the CPO detected by the cardfish. The size of the cells are quite small, about 0.2 or so. So that the E. coli is about like 2 micrometers long. So the size is very small compared to their normal bacteria. And then cells are attached to something fluffy stuffs. So when we look at SEMEDS, we found that they contain the silica, iron, and magnesium in this fluffy system. So we think that this is, when we think about this system, this is the mineral, probably a serpentine, periodite, like that kind of stuff, and the microbes attached to those minerals. So So we so far don't know about the CPL, the life strategy of CPL in the detail, but so genetic system of the CPL could be old. So when the here is like a phylogenetic analysis of liposome protein. So if the RUCA is in the middle of like a eukaryote and archaean and bacteria, then if RUCA is here, the CPO, the DPAN is really the closer to the RUCA. So it could be the older system. So we are looking at the liposomal system now. So then, and also, so I would like to talk about the acetogen here. But here is the gas composition at the Cedars. At the Cedars, it contains nitrogen and hydrogen, and the methane is in the water. So I wanted to show that I can't. Okay. So, but why acetogen? So when there is hydrogen in the CO2, so we think that the methanogen also can grow, but the reality is that the most of the organisms are acetogen. So why he has to have the acetogen? So then here is the thermodynamic calculation of acetogenesis and the methanogenesis. So when we look at the pH 11.9, the GPS is one of the springs and cedars. So the energetically the methanogenesis is favorable when we think about the system. So especially the CO2 concentration is really low at the site. So at the pH 12, the most of the CO2 is CO3 2 minus, but the 2 minus is really highly charged, so microbes cannot use it. So they need to dump the proton, expose the proton by using an ADP to make up available. So then, therefore, uh, the the even if the system had CO3 2 minus, but the required energy is higher, so the thermodynamic calculation suggested that the energetical is really not favorable. So when we think about the thermodynamics calculation based on the CO2 and N2 and produce acetate, so acetogen cannot beat methanogen. So, but when we look at the one of the microbes, it's called like we named it the Helicobacter. It was formerly named the NPLUPA2. So energy metabolism and carbon fixations like this. So here that the organism has the acetyl-CoA pathway to fix the CO2 and also for the energy metabolism. But interesting point was that this organism lacked the hydrogenase. So this organism cannot use the hydrogenase incapable of using hydrogen. And what is the reducing power for this acetogen? So then when we look at the organic and inorganic carbon transporters, but we didn't see any like a transport in this system. So therefore, this is probably acetogen, but cannot utilize the hydrogen and carbon dioxide, the substrate for this organism. So because to utilize CO2, so it's really required reducing power from the hydrogen because it requires like a reduced form of a phylloxin, which can be produced from the hydrogen by using the like electron bifurcation like system. So we think that so we can as it cannot utilize the hydrogen and carbon dioxide, and we have to think the different way. So one of the potential is that formate or carbon monoxide that can be produced when in the Fischer-Tropsch type process. So the many subinitiation site, as I mentioned, like when the CO2 is present, the methane is produced. But here a like intermediate of the Fischer-Tropsch types of synthesis. The methane is produced like Fischer-Tropsch types of synthesis, uh, synthetic phosphate, and here is an intermediate like a CO2 and a pomade and those kind of stuff. So as in the middle and as the intermediate, so the pomade and the CO uh, is produced. So then if this organism can utilize the CO to make it acetate, so that it's really an energetically favorable. So when we looked at some dynamic calculations at the pH 11.9, so that it's the minus 261 in terms of the Gibbs free energy, so can kill joule. So this is a much better in terms of the energies. But the concentration of CO in the deep subsurface is sulfur and all that is like really unsure things. But however, drill site like chromosite and contain carbon dioxide of 0.1 to 0.2 micromolar. So at the surface, we may not be able to detect the CO, but if the drilling site like a chromo and OMA detect the CO in the deep, so that should be the CO at the deep subsurface. CO could be present. So and the CO dehydrogenase, or when we look at this when CO dehydrogenase, that is like here is the anchored is not the one that came from the Cedars. So we see that the archaeal and bacterial type hybrid type of CO dehydrogenase. So that is important due to this carbon monoxide. So then a couple of different organisms. So here is the archaea and here is the bacteria, and we see like chloroflexide and the disulfur, this odaxiviator. These has also the hybrid type of CO. So these two both are like one is from rifle side and the one is from the African side, gold mine. So these are also deep subsurface microbes. So hybrid COGH is often seen and extremely limited in its carbon source. So now the NPL-EPT could be used as a CEO, but we start seeing like a different types of acetogen in the Hakuba system, and also the Cedars and the other sites. So that now we start describing like the one is the format reducing one, and also glycine reducing pathways. And also at the same time, like ongoing studies start identifying like how hydrogen format or format disproportionation, CO disproportionation, electron CO2 and glycine reduction. Those all the reaction can be occurred via acetyl-CoA pathway for the acetogen. So then acetyl-CoA pathway is is probably an answer to energy of the metabolism because acetyl-CoA pathway is the only one that the bacteria and archaea utilize system has a carbon fixation pathway. And also this is the only one so within known the 6 carbon fixation pathways, only the acetyl-CoA pathway can generate ADP. So that this is a carbon fixation pathway, and at the same time the energy can be utilized as energy metabolism. And also a symbiotic system with the organic molecule, the Serpentinized site, is kind of similar like so the CO2 can convert to the acetate. And also here is deduced by the waste like a universal last common ancestor based on the genomic information. And they also think that the acetyl-CoA pathway is an important as a looker. So acetogen is Serpentinization site. If we dig into more, then it should give us some very important information and answering the energy metabolism for life. So what we have learned so far from the Earth ecosystem, the Serpentinization site, so highly alkaline reduced water systems have been Serpentinization setting relatively isolated from the effect of photosynthesis hosts an unusual microbial community with a low cell density. So even at the analog site of earlier, so I see oceanic planets like a microbes, ocean world like a microbes that are that are not explainable from our current knowledge have been identified. And also hydrogen is not always the chemical energy for life. The hydrogen saturated the Cedars Serpentinized spring with extremely low redox potential. And carbon monoxide formant could be a ideal substrate for carbon fixation. A carbon fixation coupled with the energy metabolism in the highly reduced setting. So then and then how we can how did life emerge on the Earth ice and beyond? So so now the ocean world start detecting like hydrogen and the occurrence of serpentinization and start seeing like high molecular organic compound in the water system. So when we look, so if we can know like what kind of chemical evolution could occur in the ocean system, like chemical evolution, and perhaps like at what stages of their evolution could be occurred, and if we can know from the ocean world like Enceladus, then we can really know about like could be like a similar in the earlier the Earth. So of course it's quite different, but at some point, especially the life on the Earth like now is already like the effective photosynthesis is quite strong, and so therefore we really cannot really a true view of chemical evolution or early primordial biological system from on the Earth. But if we can explore the ocean world like this and the possibility of life in the ocean world, that could tell us the answer of the question, probably. So thank you very much. So here is acknowledgement and the funding sources.
Thank you very much, Shino. It seems, as our previous speaker said, at least on Earth, life indeed does find a way to exist in all sorts of environments that on the surface would seem somewhat inhospitable. I'll just remind everyone again to please pass on your questions using the Q&A button at the bottom of your screen. If there's an issue with that, I know one person mentioned they had a problem with that. Feel free to also post them in the chat, and I will pass them on. Shino, I had a couple of questions myself, but maybe I'll start with this one. Have we found any extremophilic life on Earth that we believe would have a high likelihood to survive if we were to pick it up and place it in another environment in our solar system, say in the oceans of Enceladus or Europa or deep beneath the surface on Mars?
So far I've never searched, tested such studies, but I think like if the life on the Earth exposed to like the Enceladus water, the life definitely can adapt to that system, I think. But if there is like a water, and if there is some, of course, the Enceladus water contain a lot of carbon, bicarbonate. And we are not sure about the phosphorus, but if it exists and it life that definitely can at least get it, even if we cannot find it the life on the current aspect life, if the life exposed to the new environment, life definitely can adapt to that system.
Thank you. I think that William Martin has a question. William, would you like to ask your question live?
Can I?
Yes, please go ahead.
Okay. Shino, can you hear me?
Yeah, I can hear you.
Okay. Wow. That was amazing. Really, through and through. My question is, Okay, so CO is really interesting. You see 1 to 2 micromolar CO in the effluent, yeah, in the liquids.
Yeah.
So my question is, could it be that mineral surfaces and native metals are covered with CO because it has a really high affinity for metals, and that maybe the bugs are somehow accessing CO bound to rocks, similar to the situation with Ken's organisms that access magnetite? That's my question. Could it be that there's a lot of CO2 on the surfaces of that system that the microbes might be able to access?
Yeah, I think like if the CO is attached to the mineral, that would be really like concentrated on the mineral. That is really ideal situation for microbes, I think. So
Okay.
yeah, is that the?
Yeah, it's just the amount of hydrogen. What do you recall? What is roughly the partial pressure of hydrogen that you see? Saturating, right?
It's saturating, already saturating at the Cedars. Yes.
Okay, so that means that there's very reducing conditions. Last question: do you see awaruite forming native in the minerals in the system?
Yes.
Yes.
At the Cedars, I'm not sure, so we have to see it. So we don't detect much about the nickel, but yeah.
Okay. Thank you.